Good day, everyone. So we're reviewing Wetzel's Limnology textbook. This is edition number 4. We're on chapter 13, which is the inorganic carbon complex. And more specifically today, we are looking at photoautotrophy and chemolithotrophy. So let's get started where we left it at yesterday, which is chemolithotrophs. These organisms, or bacteria, use the energy from the oxidation of reduced compounds such as sulfide, ammonium, iron, and hydrogen to fix inorganic carbon into organic materials. Similar to the situation for the anoxygenic photosynthetic bacteria, many of these organisms also derive some of their carbon from organic sources. The majority are aerobic, using oxygen as the electron acceptor. The common pathways include the oxidation of reduced sulfur compounds or iron-sulfur compounds such as pyrite, thiobacillus, or the oxidation of ammonium and nitrates such as nitrosomonas or nitrobacter. These oxidation pathways generate acid and reduce alkalinity. As these organisms need a source of the reduced compound and a source of oxygen, their range is limited. In stratified lakes, they are most likely to be found just above the oxycline, and in streams with high sources of reduced iron and or reduced sulfur. For example, thick mat of Begiatoa are found in the hot springs of Yellowstone Park. In the case of streams, these organisms produce organic matter at the expense of energy imported from outside the system's boundary. In stratified lakes, the situation is different because the energy that reduced the sulfate to sulfide came either from photosynthesis within the lake or in the lake's watershed. There are also some examples of anaerobic chemolithotrophs. Thiobacillus ferrooxidans has the more remarkable ability to oxidize reduced iron compounds using sulfate as the electron acceptor. In hydrogenotrophic genesis, carbon dioxide is reduced to methane using hydrogen as the electron acceptor. Oxygenic photosynthesis. Oxygenic photosynthesis is quantitatively the most important biological assimilation of inorganic carbon in freshwaters. This is the type of photosynthesis performed by all green plants, including eukaryotic algae from cyanobacteria and submerged and emergent microphytes. From the point of view of carbon assimilation alone, oxygenic photosynthesis increases the pH but has no direct effect on ANC. However, ANC is affected by the transformation of nitrogen and phosphorus during cell growth. For example, if nitrate is the soil nitrogen source, ANC is increased. If ammonium is the soil nitrogen source, ANC is decreased. This effect on ANC is usually quite small. So ANC, once again, is the acid neutralizing capacity, or also seen as alkalinity on the other side of the pH 7. Abundant physiological evidence indicates that free CO2 is most readily utilized by nearly all algae and larger aquatic plants. A number of algae and submerged macrophytes, particularly the mosses, can utilize only free CO2. Many algae and aquatic vascular plants are capable of utilizing CO2 from bicarbonate ions when free CO2 is in very low supply and bicarbonate is abundant. A few species of algae require bicarbonate and cannot grow with free CO2 alone. There is no clear evidence that algae or higher aquatic plants assimilate CO2 or carbonate directly as a carbon source. The CO2 supply problem. The biochemistry of inorganic carbon assimilation by algae and higher plants is interesting and complex. Despite the solubility of CO2 and relatively turbulent water strata of the pelagic trophogenic zone, the photosynthetic carbon supply is not very favorable. Rates of CO2 diffusion in water are 10,000 times lower than in air, and diffusive transport can be a major limitation. Much of the total carbon is in the form of bicarbonate and carbonate, which is not readily available to all aquatic plants. Rapid photosynthesis can rapidly reduce the total DIC and increase the pH of the water and shift dissociation equilibria so that dissolved carbon dioxide is nearly zero. This can happen during intense algal blooms and also in dense stands of macrophytes or periphyton. Experimental evidence on freshwater photosynthetic utilization of inorganic carbon indicate a strong relationship between physiological availability and the forms of inorganic carbon, for example. CO2 versus bicarbonate. These experimental results have been supported by a global survey on the occurrence of taxa that can photosynthesize using bicarbonate. The primary enzyme of inorganic carbon fixation in all photosynthetic oxygen-evolving organisms is ribulose 1,5-biphosphate carboxylase oxygenase, or Rubisco. This carboxylase is involved in fixing some 95% of the carbon assimilated in plants, with C3 metabolism, in which 3-phosphoglycerate is the first major product. Most algae and aquatic macrophytes have C3 biochemistry, and thus at least 95% of the carbon fix must be supplied as CO2 at the carboxylase site. The enzyme carboxydismutase is involved in the carboxylation reaction, which produces 3-phosphoglycerate as the first product of carbon fixation and uses free unhydrated CO2 as its immediate substrate. Carbonic anhydrase, an enzyme that catalyzes the reversible hydration of carbon dioxide, is found universally in photosynthetic cells of plants. Where CO2 is the carbon species entering the cell, there is no obvious biochemical role for carbonic anhydrase found in plants, although some evidence indicates that this enzyme accelerates the diffusion of CO2 and transport to sites of biochemical fixation by carboxylases. Bicarbonate ions have been implicated as a critical factor in the evolution of oxygen during photosynthesis. At high pH values and bicarbonate concentration, bicarbonate moves less effectively from binding sites to the chloroplast. When bicarbonate enters cells, carbonic anhydrase is needed to supply CO2 to Rubisco. Carbonic anhydrase activity is found in plants that cannot use bicarbonate as well as those that can, although such activity generally increases among algae and sometimes submerged macrophytes that can utilize bicarbonate. The ability to assimilate bicarbonate ions is variable among planktonic algae, macroalgae, and submerged angiosperms. When this ability does occur, an additional reaction is needed for bicarbonate assimilation that is not required for CO2 assimilation. Active bicarbonate transport followed by bicarbonate dehydration within the cytoplasm is apparently required and is coupled with a similarly active stoichiometric excretion of hydroxyl ions from the cells. Assimilated bicarbonate ions are used to produce high internal CO2 pools by action of carbonic anhydrase. When aquatic plants have similar affinities for CO2 and the bicarbonate ion, utilization of bicarbonate generally occurs when the bicarbonate concentration exceeds that of CO2 by more than 10 times. Free CO2 concentrations are about 10 to 20 micromoles at equilibrium with the atmosphere and somewhat higher, perhaps 300 micromoles, in many lakes and rivers. Most freshwaters contain bicarbonate concentration far in excess of 5 times that quantity. Equilibrium concentration of dissolved CO2, particularly in alkaline hot water lakes with a pH greater than 8.5, are inadequate to saturate photosynthesis in plants adapted to utilize bicarbonates. As these waters become more productive and in densely populated littoral zones of less productive lakes, pH is rapidly altered by metabolism on a diurnal basis. pH ranges from a low of 6 to a high of 10 or more per 24 hours, and can be associated with reduced carbon fixation and bicarbonate assimilation. Under stagnant conditions common to heavily colonized littoral zones of lakes, the shift to bicarbonate metabolism, as well as the increased pH, is often associated with a severe reduction in CO2. Stagnant layers around plant cells, algae, and tissues, macrophytes, clearly have major effects on limiting the assimilation of both CO2 and bicarbonates by actively photosynthesizing plants. Even when the plants are exposed to water movement by turbulence or sinking, a residual layer, approximately 10 to 100 micrometer thick, parallel to the surface remains. The rate of solute movement across this residual layer approximates that of molecular diffusion. Diffusion of CO2 and bicarbonate through these stagnant layers is an important rate-limiting process to both availability of CO2 and membrane transport of bicarbonate ions. Mucilage sheaths surrounding algal cells, especially among cyanobacteria, can further reduce the uptake rates of inorganic carbon. Bicarbonate assimilation in media of high pH assumes greater significance in large aquatic macrophytes that have morphologically long diffusion paths. Active transport of bicarbonate originating from the dissociation of calcium bicarbonate and the secretion of hydroxide ions results in the precipitation of calcium carbonate. Many submerged angiosperms and algae can utilize bicarbonate. Aquatic mosses are able to utilize only free CO2 and are restricted to waters of relatively low pH and abundant CO2, for example, springs, mountain streams, bogs, or shallow waters, or the lower trophogenic zone where free CO2 is higher than elsewhere in the lake. CO2 of the rhizosphere can diffuse into rooting tissues, most move to the leaves via internal gas lacunae and be fixed photosynthetically. Although this benthic source of inorganic carbon is widely utilized in submerged macrophytes, the percentage of the total carbon fixed photosynthetically from the rooting tissues is greater among small plants growing in poorly mineralized water. Moreover, many angiosperms with large intercellular gas lacunae refix the CO2 of respiration and photorespiration rather efficiently. The efficiency of refixation and the photosynthetic efficiency of carbon fixation must be highly plastic, related to, in part, to induced shift to bicarbonate assimilation, and can affect rates of net primary productions significantly. The effect of inorganic carbon concentration on primary production. It is reasonable to ask if primary production of biomass in aquatic ecosystems is ever limited by the supply of inorganic carbon. For the past 40 years or so, the answer has been a resounding no. There are several reasons for this. First, there is strong correlation between the concentration of total phosphorus and algal biomass in lakes. Second, in nearly every experiment performed in bottles, microcosms, or whole lakes and rivers, the addition of nitrogen and phosphorus results in a great increase in primary production and plant biomass. Third, as we have seen above, many algae and some higher plants can grow on bicarbonates when CO2 is in low supply. Low CO2 and its associated high pH may select for different algal species. If the low CO2 condition is sustained, fourth, CO2 is resupplied by diffusion from the atmosphere, and the rate of diffusing resupply increases as CO2 decreases. Once the pH has increased due to CO2 drawdown, the rate of resupply is increased even more by chemically enhanced diffusion. So while there may be a short-term limitations of photosynthesis due to extremely low CO2 given time, we do not expect to see inorganic carbon limit the biomass attained by primary producers. In some contrast to the long-held idea that phytoplankton biomass is not limited by the supply of inorganic carbon, several recent studies show that phytoplankton productivity and biomass does sometimes respond to increases in either partial pressure of CO2 or ANC. Jessen et al. Found that the supersaturation of CO2 often observed in lakes of the boreal region enhance both the rate of photosynthesis and the resulting algal biomass. Similarly, Cragg found the usual correlation between TP and chlorophyll A in a series of 204 Danish lakes. Splitting the data into lakes with ANC greater than 800 microequivalents and ANC less than 800 microequivalents revealed an intriguing pattern. For the same level of total phosphorus, TP, chlorophyll A was consistently higher in the higher ANC group compared to the lower ANC group. The difference was most striking at high TP. Cragg followed up with a series of microcosm experiments in which they varied both ANC and TP. The experimental results were similar to the correlation work. Primary production was about 5 times higher under the higher ANC conditions when TP was sufficient. Based on an analysis of how fast CO2 could be resupplied by the atmosphere, including chemically enhanced diffusion, Cragg found that the stimulation in photosynthesis was due to the update of bicarbonate directly. So, perhaps under conditions of nutrient sufficiency, there is a case for carbon limitation. Rising atmospheric CO2 caused marine scientists to ask what effect this might have on phytoplankton in the ocean. Experiments with marine phytoplankton subjected to different CO2 levels have shown a general increase in algal growth and an increase in the carbon to nitrogen ratio and carbon to phosphorus ratios with increasing CO2. The ocean is in the upper equilibrium. The ocean is in near equilibrium with the atmosphere. So rising atmospheric CO2 causes rising CO2 in the ocean. In freshwater systems, the concentration of CO2 in surface water ranges hugely among systems, much more so than the small effect of atmospheric increases in CO2. Urabe et al. Grew Senedismus acutus at 3 levels of CO2 to span the range recorded in most abundant freshwaters. Partial pressure of CO2 of 360 microatmosphere, 1500 microatmosphere, and 3000 microatmosphere. Algal biomass was 7 and 8 and 10 times higher in the elevated CO2 treatment compared to the ambient atmospheric treatment. Further, the CP ratio increased across the CO2 treatment from about 285 in the ambient treatment to about 1000 in both of the elevated CO2 treatments. Urabe et al. Concluded that CO2 limited the growth of this algae in culture. Interestingly, the high CO2 algae were a proper source of food for Daphnia than those grown at ambient CO2. Vogt et al. Surveyed 69 boreal lakes that varied in partial pressure of CO2 from undersaturation to about 2,000 microatmosphere and found that both chlorophyll A and gross primary production, GPP, increased along the P-CO2 gradient. For GPP, there was a nearly linear increase across the P-CO2 range. For chlorophyll, the relationship only held true for those lakes, most of the ones in which this data set is from, that were above atmospheric levels. That is, in the very low CO2 lakes, chlorophyll A, but not GBP was high. In summary, in typical freshwater, DIC is dominated by the bicarbonate anion, and this carbon pool is usually as large or larger than that of dissolved organic carbon particulate, organic carbon, and planktonic organisms. The major exception is soft acidic boggy waters, where DOC is high and DIC can be quite low. DIC concentration range from as low as 10 micromoles per liter to more than 5,000 micromoles per liter in typical lakes and rivers, although there are places where it can be much higher, such as lakes in the karst regions or at the bottom of deep lakes with underlying volcanism. The complex equilibrium reaction of inorganic carbon and the distribution of the chemical species of total dissolved inorganic have been well understood in considerable detail for some time. The DIC system is the major regulator of both the pH and buffering capacity of typical freshwater ecosystems. DIC is equal to carbon dioxide plus bicarbonate plus carbonate. The pH of the water is largely controlled by the ratios of the species of DIC. As carbonic acid percolates over rock and through soils, carbonates and silicates are solubilized. Ionized calcium and bicarbonates are released into the water. The dilute solution of bicarbonates of many freshwaters is weakly alkaline because slightly greater concentration of hydroxide ions than protons ion or H+ ions results from the dissociation of bicarbonate, carbonates, and carbonic acid. The concentration of DIC tends to be highest in carbonate geology, but at the global scale, the delivery of DIC to the ocean is nearly equal from carbonate and silicate geological regions. This is due to the very large hydrologic flux of moderately high DIC from the humid tropics, where silicates or basalt geology often dominates. Losses of CO2 by photosynthetic utilization or addition of CO2 from biotic respiration are the major factors that change the pH, H+ ion activity, of a given water body over time. CO2 and pH vary inversely over diel and seasonal cycles. The buffering capacity, or ANC, of the water, however, tends to resist changes in pH as long as the equilibria of the DIC complex are operational. The distribution of DIC and pH in surface waters vary both seasonally and vertically in lakes in relation to a loading from allochthonous sources, physical conditions, and biotic inputs and consumption. Alkalinity often increases in the anaerobic portion of stratified system due to anaerobic respiratory processes such as sulfate reduction. Most lakes, rivers, and streams are supersaturated in terms of CO2. Thus, there is a constant net efflux of excess CO2 from most inland waters. This net CO2 evasion to the atmosphere derives from both inputs of CO2 in groundwater from the watershed and an excess of respiration over gross primary production within the water body. Net heterotrophy is supported by the input and respiration of terrestrial organic matter. In thermally stratified lakes of low to moderate productivity with minimal inputs of organic matter from outside the basin, the accumulation of CO2 in the hypolimnion is positively correlated to rates of organic production in the trophogenic zone. Inorganic carbon is a major nutrient of photosynthetic metabolism. However, phosphorus and nitrogen limit photosynthesis more frequently than does inorganic carbon, which occurs in much greater abundance. Because assimilation of CO2 and bicarbonate occurs more rapidly than resupply, and conditions of reduced availability results from diffusion resistance at the uptake sites, potential photosynthetic productivity by both algae and submerged macrophytes is not always fully realized. Many algae and vascular aquatic plants have developed compensatory mechanisms to enhance the utilization and recycling of respired CO2. And that will be it for today. And sorry for being a little longer, but I wanted to go through the summary, and tomorrow we'll start with chapter 14, and let's look it up. And I believe it's going to be about the nitrogen cycle, so but let's confirm that. Yeah, the nitrogen cycle, chapter 14. So I'll see you tomorrow. Thank you, and I'll talk to you soon. Bye.